Hello everyone and welcome to today's webinar, Transform Your Bioanalytical Assay Development with TrailBlazer Antibodies. My name is Freya Leask, I'm the publisher of Bioanalysis Zone and I'll be your host for today's event. Before I introduce you to our speaker, I'd like to quickly cover a few housekeeping items. So all of the widgets on your screen are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right-hand corner. A copy of today's slide deck and our feedback survey are located in the resource list, and we encourage you to download any resources or links that you may find useful. Please do let us know your thoughts on today's event by tweeting us at Bioanalysis Zone or by using the hashtag BZWebinars. Or you can simply share today's webinar with your friends and colleagues using the Share This widget on your screen. Some networks can cause slides to advance more slowly than others, so we'd recommend logging off any VPNs. And if your slides are behind, you can push F5 on your keyboard to refresh the page. You can find additional answers to some common technical issues located in the Help widget at the bottom of your screen, and an on-demand version of today's webinar will be available around 1 hour after the webinar, and it can be accessed using the same audience link that was sent to you earlier. So I am delighted to introduce Paul Royal. He's a technical sales manager for custom antibodies at Bio-Rad Laboratories in the UK, who's presenting for us today. Paul is a technical sales manager for Bio-Rad's custom antibody service, and he's been with Bio-Rad's antibody division for over 7 years. Prior to this, he worked in an immunology diagnostics company for around 7 years, and Paul holds a degree and PhD from the University of Nottingham and has postdoctoral research experience from the University of Warwick. So I'd like to invite you, our audience, to submit any questions you may have for our presenter throughout the webinar using the Q&A widget to the right-hand side of your screen. And I'll be posing your questions to Paul live after his presentation, and then we'll follow up any outstanding questions offline. So thank you again for joining us today, everyone. I'll now hand over to Paul to start today's presentation. Over to you, Paul.
Thank you to the organisers. My name's Paul Royal and I'm here to talk to you today about our TrailBlazer antibodies. So these are new features we've added to our custom antibody service which give increased flexibility when it comes to the formats of the antibody we produce. In this presentation I'd like to cover first of all our custom antibody service and the method by which we generate custom antibodies. I'd like to give some bioanalysis relevant examples of how we can generate highly specific antibodies using this technology. Then I'd like to cover the SpyTag technology, which is the technology which underpins the TrailBlazer antibody products. And finally show the utility of this technology in relation to bioanalytical assays and bioanalytical studies. The Bio-Rad custom antibody service is based on the HuCAL library. So the HuCAL library is a gene library, and the genes encode the fragments of antigen binding of fully human antibodies. HuCAL stands for Human Combinatorial Antibody Library. This slide really shows what the gene library looks like at the DNA level. So you have the genes encoding both the variable, heavy, and the variable light chain regions of the antibody. So in terms of diversity, you have diversity at the level of framework. So you have 7 variable heavy master genes and you have 6 variable light master genes. 3 of them are kappa, 3 of them are lambda for the light chains. So there are 42 framework combinations before we even start. The framework genes themselves have restriction sites into which pre-built CDR cassettes, so CDR is complementarity determining region cassettes, or the cassettes encoding the hypervariable regions of the antibody. But these are inserted into the frameworks using these restriction sites. And then there is diversity at the level of the CDRs, which are diversified by a process called trinucleotide mutagenesis. So it's a fully synthetic library, and there's no particular immunological bias. The library has undergone no process of tolerization, and the library is not in any way similar to an amplified immune repertoire. So the nature of the library is such that it's just a really very diverse library. There are approximately 45 billion different antibodies in the library, so the chances are the antibodies the customer requires already exist in the library and have always existed in the library, We simply need to devise a strategy to go into the library and identify the antibodies which meet the customer's specificity requirements. In order to do this, we use a technology called Phage Display. At its most simple, Phage Display is a link between genotype and phenotype. In this case, it's a link between a functionally binding antibody fragment and the gene that encodes that fragment. So the protein itself is displayed upon the surface of a phage, and within the phage-mid, the phage genome, is the gene for that particular antibody fragment. Now, as I said, these proteins are functional, so what Phage Display enables us to do is use a process of enrichment called biopanning, whereby the antigen of interest, or the customer's protein target or target of interest, is immobilized. The entire Phage Display library is added to the immobilized antigen. The non-binders are washed away, the binders are recovered, they are then propagated, harvested, and the process is repeated over 3 rounds. And the net effect of this process is to take your incredibly diverse library from a very large non-biased library all the way down to a much smaller library which is full of binders which are highly enriched for phage displaying antibody fragments which recognize that customer protein of interest. A key advantage of using a fully in vitro method for generating antibodies is that the library generation process and conditions can be manipulated to achieve antibodies that meet very high specificity requirements. So there are various strategies that we can use, and in this example I'm showing the blocking strategy. So to describe this in a context of a bioanalysis relevant example, imagine that the analyte is a human therapeutic protein that is being measured during a preclinical study in the serum of a non-human primate. And that non-human primate might have a homologous protein which is, say, 95% homologous by sequence. So the challenge is very much to generate antibodies that can discriminate between the human version of the protein and the primate version. So in terms of how we would do this, we would immobilize the target antigen, the human antigen in this case, shown in blue, and we would block in the solution phase with a higher concentration of the closely related antigen, shown in white. So this would be the primate homologue of that protein. And we would use a higher concentration of the blocking protein so that any cross-reactive antibodies are absorbed out over the 3 sequential panning rounds, so that only those phage displaying antibodies which recognize features unique to the target antigen are enriched. Using the blocking strategy and other similar strategies, we are able to generate antibodies of very high specificity. One area where we have a lot of expertise is in generating antibodies that recognize therapeutic drug antibodies. So these are known as anti-idiotype antibodies. So in the diagram here, you see the therapeutic drug antibody in gold, and you see the anti-idiotype shown in purple. So we are able to use the conditions of the library not just to generate anti-idiotypes generally, but to actually generate different classes of anti-idiotypes. So I'll describe how we generate them in the next slide, but in this one, for the first 3, certainly I want to describe the different classes of anti-idiotypes that we commonly make. The first type is the type 1 anti-idiotype. So here you see the anti-idiotype is binding to the antigen binding site, or the paratope, of the drug antibody. So these antibodies are useful if you need to measure free drug in a human serum sample, and for anti-drug antibody controls if you need ADA controls that are neutralizing to the drug, i.e. antibodies that obstruct or interfere with the mechanism of the drug antibody binding to its target. The next type we make are the type 2 anti-idiotypes. So here the challenge is to identify unique epitopes to the drug antibody that are outside of the paratope. So these are non-inhibitory antibodies. So these will allow you to measure total drug, regardless of whether the drug is free or in complex with its target. And this can often be a requirement because often the target of therapeutic drugs are present in the serum. So when a sample is taken, the drug antibodies that are recovered will be in complex with their target. They will not be in the free form. Then we have the type 3 anti-idiotype. So here the anti-idiotype only recognizes the drug in complex with its target. And This isn't just true for drug-antibody, drug-target complexes. These can be any protein-protein interactions. But here, essentially, the challenge is to generate antibodies which recognize the complex but don't recognize either component of the complex in the free form. Finally, I've added an additional example. So this is going slightly away from antibodies to Fc fusion proteins. And in the example I've given here, we have the example of etanercept. So we've made this as a catalog antibody, and essentially the challenge here is that etanercept is a TNF-alpha receptor, Fc fusion protein, and there's a form of the soluble TNF-alpha receptor in the serum, and obviously there's Fc in the serum with normal immunoglobulin. So the challenge here was to generate antibodies to a unique feature on this particular drug, and that unique feature is the junction between the receptor and the Fc domain. And I'll just briefly describe the process we did to generate this. So we screened our library on etanercept, and we simultaneously blocked with soluble TNF-alpha receptor and human Fc, so that we only got antibodies that recognize that junction, which is unique to the drug. In terms of how we generate the anti-idiotypes given in the first three examples in the previous slide, this slide illustrates the panning methods we use. So there are different panning methods to generate inhibitory and non-inhibitory antibodies. So for inhibitory, it's quite simple. The drug antibody is immobilized, as shown in gold on the bead in this case, and isotype controls, either germline or framework matched or human serum or both, are added in the solution phase, so that only antibodies which recognize something unique to the drug antibody, such as the paratope, are captured, whereas those which recognize common features, such as the Fc domain, are blocked away, as I demonstrated in the blocking mechanism, and are lost during the wash step. For non-inhibitory antibodies, rather than coating the solid surface with the drug, we coat the solid surface with the drug target, and we use a high concentration of drug target and then a lower concentration of the drug antibody to give ourselves confidence that all of the paratopes in the drug antibodies are occupied. We then block with isotype controls and human serum again, and also with the drug target to make sure that we don't generate antibodies to the drug target. By this method, this method is used in the generation of both type 2 and 3, but the difference is for type 2 anti-idiotype generation, where you want antibodies which recognize both the free and bound form, we alternate between inhibitory and non-inhibitory, so that you generate antibodies that are outside but recognize both. And for the complex specific, we screen only for non-inhibitory antibodies whilst blocking with both the components of the complex in an alternating blocking strategy. After biopanning, shown in this schematic representation of our process as guided selection, there is a subcloning step. This is a slide showing our old process, and this really was where there was a limitation in terms of the flexibility we could offer. So we could offer the customer choice in terms of final format. They would select the format they wanted, and we would subclone into the appropriate destination vector. But any change of format thereafter was either achieved by subcloning, a process which takes 6 to 8 weeks, or by chemically modifying the Fabs, such as by conjugating to HRP or fluorophore. Now we have a new technology which underpins the TrailBlazer product range called SpyTag. And what SpyTag enables us to do is add further flexibility to the formats that we can deliver at the end of the project. For many of our projects, the process now looks more like this. So we tend to subclone into a monovalent Fab expression vector, which produces the Fab with, amongst other things, a 13 amino acid SpyTag on the C term of the heavy chain of the Fab. And one of the reasons for this is that the monovalent Fabs are a good format for using in K-off ranking or any kind of affinity work, because with a monovalent format you get a nice clean signal when you're studying affinity. When you have bivalent formats or multivalent formats, you have an avidity effect. So avidity is a function of both the affinity of the binding site and the number of binding sites in that particular format of the molecule. So historically, customers who wanted a high affinity antibody, they'd be at a sort of crossroads. Do they choose the monovalent format so that they can do affinity work with the Fab and so that they can sort the high affinity Fabs from the lower affinity Fabs? Or do they get the higher overall binding strength format, the higher avidity format, and get the bivalent from the off, but then not have the option of measuring affinity as accurately? With the SpyTag technology, what we can now do is we can make the antibodies in the monovalent format, identify the high affinity binders, and then rapidly convert those into a bivalent format to get the avidity benefits before delivering them to the customer. So I'll explain a little bit more about this SpyTag, which we have incorporated into our Fabs in the next slide. SpyTag gets its name from strep pyogenes. So within strep pyogenes is a fibronectin binding domain that contains a collagen adhesion domain, and within this there is a bond between the lysine at position 31 and the aspartic acid at position 117. And what this bond is a peptide bond, it's an amide bond, it's a covalent bond, and it forms spontaneously. The reaction energy comes from that glutamic acid at position 77. And when this domain is split out into the 13 amino acid SpyTag and the rest, which is called the SpyCatcher, this bond will still spontaneously form. So really what you have is a method of spontaneously ligating two different proteins, the SpyTag protein with the catcher domain and anything else built onto that catcher domain. And this is really the technology that we've utilized in our TrailBlazer antibodies. So whereas previously we could only offer format conversion at the DNA subcloning level, now we can offer format conversion at the protein level as well. And we refer to this internally as protein ligation. For the subsequent slides, we're going to use a more simplified version of the diagram you saw previously, represented here. So what you see is the 13 amino acid SpyTag and the 15.2 kilodalton SpyCatcher, and then the image on the right is what they look like in the ligated form. As mentioned in the previous slide, this is a spontaneous reaction. The bond is covalent, it's an irreversible reaction. It's very fast, we leave it for an hour, but in reality it's done in minutes. And the resulting protein is very robust. This reaction can even occur within cells. By choosing to have the antibodies in a SpyTag format, the customer now has the choice at any point after their project of changing the format of the antibodies they've received or receiving the antibodies in multiple different formats according to their requirements. So for example, they could have single SpyCatcher domains which can be used to attach different labels to the antibodies. There can be bivalent bycatchers to dimerize two Fabs to get the avidity benefit, or there can be FC catchers if you want to get some kind of serological control. The monovalent and bivalent formats can be naked, or they can be coupled to biotin or peroxidase. And the FC domains, again, can be naked or coupled. And when it comes to the FC domains, we don't just have different human immunoglobulin classes and subclasses, we also have different species FC domains. This can be particularly useful if you're making primary antibodies, for example, to fit in with immunostaining panels where you have predefined or optimized secondary detection systems that you want your antibodies to slot into. So we see a lot of possibility with this technology in that area. When it comes to labeling, we've introduced cysteines into some of our SpyCatcher domains, and this allows us to achieve site-specific labeling using maleimide chemistry. So what this gives us is a fixed degree of labeling, which in turn gives high batch-to-batch consistency. So not only does this give high batch-to-batch consistency, but imagine a scenario where we've done a custom project, we've generated 15 antibodies. If you wanted to use the SpyTag-SpyCatcher technology to produce coupled versions of those antibodies, all of the couplings would have been taken from the same pre-labeled batch of catcher. So each of those 15 antibodies that we generated for that project would have exactly the same degree of labeling. So when you're evaluating all those different hits to choose which ones you want to progress with in your assay, you'd be confident that the differences you're seeing between different clones would be down to the performance of the binding characteristics of that antibody rather than any difference in labeling, which you may see if you labeled, say, 15 different antibodies by a chemical method such as primary amine coupling. Another advantage is whether you're using the site-specifically conjugated SpyCatchers, such as those shown, or whether you're using a format where, say, a human FC has been labeled using a primary amine type chemistry method. All of that labeling has occurred before the Fabs are added, so you can be confident when you're using the Fabs that or when you're using the whole antibody, that there will be none of the label will be incorporated into the paratopes or the antigen binding sites of your antibody, where they might negatively affect the binding characteristics, because all of the sort of chemical labeling will have occurred on the catcher proteins or related proteins before the Fabs were even added. Here we have used essentially direct ELISAs to compare different antibody formats of the same antibody. So we have two different SpyCatcher domains, they're all labeled, and we also have a mammalian expressed version of the same clone, which has been labeled by a primary amine chemistry method. So you see the different schematics on the right, and on the left you see the performance, and the version that was made by mammalian expression is the trend line is shown in blue. So you see we achieved, when we compared them directly, we saw lower sensitivity by the method where we'd used primary amine coupling rather than coupling to pre-labeled catchers, whether that was site-specific as shown in the bycatcher HRP, or whether it was an FC catcher which was labeled by primary amine coupling prior to adding the Fab domains. In terms of interpreting these results, we assume that there must be some modification to some of the antigen binding domains of the mammalian expressed antibody, because we certainly seem to achieve better sensitivity with the SpyTag versions. To give a bioanalysis relevant example of how these different Fab formats compare, what we're showing here is our standard pharmacokinetic assay setup. So you have a monovalent capture. The idea is you can coat the monovalent antibody thinly enough that only one binding arm of the drug antibody shown in gold is captured, leaving the other binding arm free for detection. And then we typically favor a bivalent capture because it has a slower off rate. Now I should point out in this slide that the detection antibodies, we used fourfold less of the catcher formats than we did of the mammalian expressed HRP to achieve this kind of equivalence, because of the difference in sensitivity you saw in the previous slide. But essentially what this slide shows is that you can get very high equivalence in a sort of ligand binding assay setup using the SpyTag technology to make bivalent formats of the Fab compared to a fully human IgG expressed in a mammalian system. Sticking with the pharmacokinetic assay example, I want to deviate slightly from the SpyTag TrailBlazer antibody story at this point, because not all drug antibodies are intact IgGs. We're increasingly seeing a number of therapeutic antibodies that are antibody fragments, and this can present certain difficulties when it comes to designing pharmacokinetic assays to detect them, because you don't have one binding arm free for capture and one for detection. You only have one binding arm overall. And one of the ways we overcame this was with the catalogue antibodies to the drug Ranibizumab. So Ranibizumab is a 48 kilodalton recombinant human antibody which is specific to vascular endothelial growth factor A. So to develop pharmacokinetic type assay formats for the measurement of Ranibizumab, we combined our type 2 and type 3, our non-inhibitory anti-idiotypes, with a capture format to develop different assays. So essentially in both assays we coated our solid surface with the drug target of Ranibizumab, the VEGF-A, and we allowed then the Ranibizumab from the sample to bind, and then we detected with our non-inhibitory antibody whether that was a type 2 or a type 3, but essentially that was the labeled detection antibody, and in both of those cases we were able to develop sort of good titration curves for the measurement of ranibizumab, free ranibizumab in the sample. So from pharmacokinetic assays back to a different type of SpyTag format comparison study, again keeping in the theme of the bioanalytical world, what we're looking at in this slide is anti-drug antibody assays. So here you've developed a drug, you need a positive control material to simulate an immune response to your drug, but obviously there aren't many patients with that because nobody's ever been exposed to a molecule you've just developed. So here our anti-idiotypes can be used as serological controls to simulate that immune response. In these two graphs, what we have are three different Fab clones. So we have basically a high affinity anti-drug antibody, a medium affinity anti-drug antibody, and a low affinity anti-drug antibody. And we have those three clones both in a mammalian expressed full IgG format in the right-hand panel, and on the left we have those as the Fab format in our SpyCatcher coupled FC panel. And what you see here is very good equivalence, and what you also see is that the characteristics, the binding strength of those antibodies haven't been significantly affected by having the catcher domains rather than the full mammalian format conversion. Our format comparison studies aren't limited just to ELISA-based techniques. We've done comparisons with a number of different assay formats, including Western blotting. Here what you see is really a comparison between our SpyTag-SpyCatcher bivalent format, either used naked as a primary antibody and then with secondary detection, or directly labeled using the site-specific maleimide HRP conjugation. And what you see in this study quite clearly is that a better degree of sensitivity is achieved when you're using the directly labeled antibody rather than using the naked version as a primary antibody. We see the utility of the SpyTag-SpyCatcher technology extending beyond just rapidly switching between different antibody formats. We are currently looking at a number of different applications for this, including the use of coating solid surfaces, be that microtiter plates or beads with SpyCatcher, to give orientated, rapid and controlled immobilization of Fabs, to use as capture reagents, also as for rapid capture to resins for affinity columns. Another area we're quite interested in exploring as well is the use of this to make simple bispecific antibodies for sort of early prototyping. Here the idea would be that you make one of the Fabs in an expression cassette featuring the SpyTag, a different Fab would be made in an expression cassette featuring the catcher, and then when you mix these two Fabs together you would get one-to-one coupling, and the result would be essentially a bivalent antibody. So I think there's a lot of exciting developments coming. I hope you continue to follow us. I'm sure we'll make it worth your while. So this brings me towards the end of my presentation. I hope I've given you a quick insight into the custom antibody service we offer using the HuCAL Platinum Phage Display system. I hope I've given you some examples, maybe not just of anti-idiotype antibodies, but antibodies that can discriminate between human proteins in a primate serum sample or antibodies to Fc fusion proteins. I hope I've generated some interest with the SpyTag technology, showing how it can give versatility and flexibility at the protein level for antibody formats, so that you now have flexibility both at the DNA cloning level and the protein level, and how these antibodies can be useful so that you can receive your antibodies in multiple different formats for multiple different applications and evaluate them all simultaneously. We do have a lot more information on this technology. You can visit our website or contact me or one of our HuCAL technical sales specialists. We're always happy to give more in-depth descriptions of our technology and talk about specific projects you may be interested in. So, yeah, with that, I would like to take any questions, and thank you to the organizers, and thank you all for your attention.
Fantastic. Thank you so much, Paul, for that wonderful presentation. So I would now like to move on to the question and answer section of today's session. So you, our wonderful audience, still have time to submit any questions that you may have for Paul using the Q&A widget to the right-hand side of your screen. So we do have time to pose some of the questions that we've received to Paul so far. So our first question is, can SpyTag antibodies be used in vivo, Paul?
We don't believe, well, we didn't really envisage them being used in vivo when we generated them. To explain why, the Fab part of the antibody is E. coli expressed. So when we make it, there's going to be endotoxin present. And even when we mix it with a mammalian expressed, say, Fc capture, some of that some of that endotoxin will be carried over. So the other thing is the catcher domains are 15 kilodaltons, and obviously their origin is strep pyogenes. So we more envisage the TrailBlazer SpyTag antibodies to be used in ligand binding assays rather than in vivo, because we'd be worried about the endotoxin having other effects.
Fantastic, thank you. Our next question is, can I add a different label to a SpyCatcher myself?
Yes, so the different capture domains, the uncoupled naked capture domains, aren't available in the catalogue yet, but we're planning on putting them in. We're also planning on making lots of different formats so the choice will increase. But you can buy them off us directly as sort of part of the custom service. So if you do want them either the sort of naked normal versions or with the site-specific cysteines in for site-specific conjugation, you can just buy those a milligram at a time, and then when you buy the Fabs, you can do the coupling yourself. So yes, they are available, not on the website, but as a custom at the moment, and they will be on the website soon.
Fantastic, thank you very much. Okay, so unfortunately, I think that we're just about running out of time, but thank you so much for those fantastic questions. Paul, is there anything else that you would like to add to our audience before we wrap up?
Not really. Thank you, Freya, and thank you, the other organisers. And if anybody has any questions, I'm sure you can get my details off the website, so feel free to get in touch, and we're always here to help. So yeah, thanks.
Fantastic, thank you. So absolutely, as Paul has said, he will do his best to follow up with questions. And to all the audience that sent questions that we didn't have time to answer, we will do our very best to follow up with those for you.
Okay.
So, thank you very much, Paul, for your time, your fantastic presentation, and your time answering the questions. Thank you again to our wonderful audience for posing your fantastic questions and your time today. We'd love to know your thoughts on Paul's presentation, so please do complete our feedback survey, which you can find in the resource widget, or tweet us at BioAnalysisZone. And don't forget to visit bioanalysiszone.com for more webinars. So thank you again for attending, and goodbye.